Have you ever wondered what the building blocks of you as a human being are? You're about to meet a man this morning that really has made his career, his profession, his passion, and looking for those building blocks down the molecule and DNA of you as a human being, and uses it for the good and tries to find the cure for cancer and many other ailments from a neurological point of view. So I want to introduce you to the CEO of Brain Institute of America. Let me bring on Brian. Hold on.
Is a devastating medical condition that affects millions.
Let's see. Is that the one? There we go.
Is a devastating medical condition that affects millions of people worldwide. It is often described as being a prisoner in your own body because stroke can lead to paralysis, loss of speech, and other cognitive abilities. Stroke survivors may feel trapped and helpless as they struggle to perform simple tasks we take for granted. Physical therapy, speech therapy, and occupational therapy can help stroke survivors regain their independence. It's important to recognize the signs of stroke and seek immediate medical attention because time is of the essence when it comes to treating this condition. Prevention is key, so adopting a healthy lifestyle can potentially reduce the risk of stroke. Stroke TV Media is here to help you and your loved ones rebuild your life after stroke.
Well, good morning, Brian.
Good morning, sir. How are you?
I see you got your background working. Looks great. Looks really good.
We're trying over here.
You know, it's so funny because Brian and I just had both our backgrounds messing up. And we're going to go with it. So if we blink in and out a bit, that's the way it is. We're here for the content. And if this is broadcast live on YouTube, and also broadcast will be uploaded to a podcast only, which is audio only. So, Brian, introduce yourself. Welcome.
Well, thank you for having me on the show. Some people know who I am, some don't. This is a very wonderful opportunity. My name is Brian Adams. I'm president and CEO of the Brain Institute of America. My company has been around since 2016, 2017, formally. We incorporated back then. And primarily we work on different types of cancers and other neurologic disorders. My training is a molecular biologist, so we work on something called non-coding RNA. To put it in perspective, back in the late 90s, early 2000s, we did the Human Genome Project. We found there's about 26,000 or so protein coding genes in a human being. And that was great back then, and it held the promise for discovering the genetic basis for a lot of disorders, and we've succeeded in that mission. There's been quite a few therapies out there from that project, but everyone kind of forgot about the other genetic input, and that's non-coding RNA. And I don't want to bore the reviewers too much on the science side of things, but just ribonucleic acid, or RNA, takes on different forms. It basically helps the body make protein. It translates the message of the genetic code, and certain RNAs, like the ones I work on, these smaller RNAs, they actually regulate the abundance of like how much the gene is being expressed. So how blue are your eyes? How dark is your hair color? If there's a gene that's causing cancer, how much is it being expressed in your body at one time? And so those, were kind of forgotten about for quite some time. Probably some people have been working on RNA for a while, but it wasn't really a well-established field or kind of well-studied back then. I'd say probably in the last decade, We've had thousands of papers on non-coding RNAs, and they make up about somewhere on the order of like 100,000 different genes are non-coding RNA. So that's, we're talking 3 to 4 times more genetic information than we thought we should be dealing with back in, compared to the 90s. And so I was fascinated by that, growing up and being in grad school, and during my academic studies, we found a couple of small microRNAs actually that regulated estrogen receptor in breast cancer, surprisingly enough, and tried to study a bit of those. And I think throughout my academic career, I just got really interested in understanding how the neurologic system worked. It's a very complex organ site, and there's a lot of neurologic disorders that we don't really have, any understanding of how things work. We kind of have a little better understanding in the past 5-10 years, but probably 2 decades ago, kind of clueless about what was going on. So we've been applying the microRNA technology I just talked about, these small RNAs that regulate the abundance of genetics, the genetic information. And we've been trying to see if they are going to work as a therapy or a biomarker for things like Alzheimer's, Parkinson's, maybe not really a biomarker for stroke, but more of a therapy for stroke, help in recovery in that sense. And it makes sense. It makes perfect sense. These are small RNAs that regulate a lot of different genes at once, and a lot of the disorders I just mentioned, it's multi-genetic. So it's not just one gene causes the problem. Many different genes actually cause the problem. So we've actually found some cool microRNAs that target many different genes all at once, and they kind of work together to kind of control normal cell function, restore normal cell proliferation, control neuronal development, things of that nature. So yeah, I mean, it's a fascinating time for anybody who's interested in being in the RNA field. That's definitely for sure. For our group, we've been developing a few therapeutics over time. We're trying to work on some delivery strategies, make sure it gets to the right organ site. Takes a little while to do that. So we've been working on those for a while. And then, I've just been dabbling in a few other things in the meanwhile while we're trying to get that pipeline up and running. We survived COVID, and we got ourselves involved in some educational projects. We've been trying to help some students get their careers launched. And we also started a nonprofit to actually support and be advocates for individuals that suffer from these neurologic disorders. So yeah, this is kind of where we've been for the past 6-7 years.
So Brian, let me ask you, guess more of a just I'm trying to grab my head around this whole thing. First of all, what does RNA? It sounds like an abbreviation. What does it stand for?
Yeah. So to answer that question, I'll back up and say what DNA is. So when everyone, you hear a news report about some genetic thing in the DNA, or that the DNA is your blueprint, I mean, it's just a sequence of code. Letters A, T, C, and G. Just a chemical, the nucleic acid, and the sequence of those letters actually will eventually code for a certain protein. Make a long story short.
But you can actually see, let's just take cancer. You could see I don't know, I mean, it's our term, but you could see the gene, the RNA, the cancer gene, and you can modify that with the RNA.
So there's a couple of questions. There's a couple of things going on there. So first, yes, we can sequence the genome of a tumor or a patient. We actually do that. That's actually established in the field. That was a big push in the past 5 to 10 years. We could do a whole genome sequencing, so I can know your entire genetic sequence of you. And if you have a tumor, I can know the entire genetic sequence of your tumor. What that means is, tough sometimes because it's a lot of information, and we've pretty much studied only a subset of genetic code, so we can probably interpret only some of that information. So this is like big bioinformatics data that we're talking about with those types of technologies. But that's DNA. And so normally when we were taught in school, RNA is just a modified form of DNA. It's ribonucleic acid, not deoxyribonucleic acid. So long story short, it's a little less stable. It's more reactionary because of the way the sugar molecule is on the RNA. But when we were in school, it was always taught that DNA makes messenger RNA. And the reason why it does this is DNA is bulky. It's in the nucleus. It's just the roadmap to like the genetic information about what makes you you. And all of the stuff that makes the protein, all the enzymes, all the structural proteins, that's in the cytoplasm. And so the genetic code kind of gets transferred from DNA to messenger RNA, and the messenger RNA goes to the cytoplasm, and the ribosomes basically make the protein. So what happened for a really long time was that messenger RNA, or mRNA, was kind of relegated as just the carrier of information, and nobody really cared about it because, you really just wanted to target the DNA, because if we found a mutation in your genetics, we just want to target that, right? We don't need to care about the message. But when we started doing and uncovering more biological processes, we found that there's a whole subset of genetic information that's transcribed. It makes what looks like to be a messenger RNA. It makes a long RNA, and then, but instead of it being made into a protein, it stops. It gets processed by a bunch of proteins in the nucleus, and you end up with these small little 22 nucleotide microRNAs that actually bind to and kind of regulate the abundance of that messenger RNA that I talked about. So that's kind of where this whole idea of the abundance, so even of a gene, is important. So even if maybe you didn't have a mutation in an oncogene, because we always thought about this in the DNA. Maybe you didn't have a mutation at all, but you have an overabundance of that gene. It could still cause cancer. So it's sort of a way of, a different way of looking at the genetic landscape of a cell, and how it can become basically a faulty cell. Now, what you're also talking about, go ahead.
For the average person, this is deep. This
I
is
know.
so deep. This is so deep, but I think I'm grasping a little bit of it. So, if you see the cancer gene, do you have the ability to go in and remove it?
There are technologies out there that we can do this. In patients, it's limited. It's not. We probably have a couple of therapies on the cancer side. There's a few companies that have developed RNA-based therapies that can edit the DNA. So that's kind of why I wanted to take a step back and explain, first of all, what's DNA, and then what's RNA, and then what I work on, which is microRNA. That's the first step, because when we start talking about gene editing, and how RNA can do this, it's also a whole different question, because what's going on there is almost akin to what we call CRISPR. But basically,
CRISPR was very similar. CRISPR cuts it out or something.
Yeah, there you go. Yeah, so basically, it's RNA. So basically, it shows you that RNA just doesn't bind to RNA. RNA also regulates the DNA. And RNA can recruit a bunch of proteins that bind to DNA itself and can actually edit the DNA, or block it from being transcribed. whatever it may be. But that's one way to fix a mutation for sure. If you can get an RNA into the right cell, and you can find the right sequence, you can force the body to do a certain type of DNA repair. We'll put it that way. Make it very simple. So
Well, you're saying that
yeah.
if you identify the cancer gene, then
Well, there's going to be multiple cancer genes. I think that's the thing. That's the first problem that we have. That we always, you're always thinking it's a cancer gene, or maybe something that kind of, in this case, for your individuals that have stroke, maybe it's something that causes blood vessels to be really leaky, right? And you kind of want to repair that process. So, first you have to find the right, well, the major gene that may be causing that problem, or a series of genes that are causing that problem. That's the first issue. There's probably not just one smoking gun. I think that's actually what makes the research side a little bit more cumbersome. Takes a little longer to figure out what's going on. There are some. So let's say you're in your case, there are some rare types of cancers that are literally have occurred because you had a chromosomal fusion. Like literally like you have a certain set of chromosomes, they're all normal. You have 23 pairs of chromosomes or whatever. A piece of a chromosome, okay, didn't replicate right and stitched onto another chromosome that it's not supposed to. So those are called gene fusions, and they make some really bizarre kind of genetic proteins, right? And so the long story short of that is we can target that pretty easily because it's so abnormal and out of the box that your therapy is not going to target anything else, right? It's going to go right towards those fused proteins. And there's really good therapies that treat those patients and cure them. We've done a really good job at that. I think the metastatic cancers, by the time a breast cancer becomes metastatic, by the time a liver cancer becomes metastatic, by the time you get grade 4 glioblastoma, there's so many different genetic pathways that are disrupted that it's just really tough to use just one therapy and hope that you're really going to, cure that patient. Which is actually why the field has gone to what they call personalized medicine. They're trying to get to this point of you go into the clinic, you have a tumor, they can sequence your tumor and understand as much as they can about what is wrong, like what's genetically driving that tumor. And the idea is to maybe put that patient on a couple of different therapies. That's, how successful that's going to be, we'll have to see. This is an idea that's kind of been rekindled in the past 5 to 10 years. So it's going to take a little time to figure out all of that. But it's a smart way of thinking about it. I mean,
so you're saying, I'm focusing on saying cancer a lot, but now reality, you're focusing also on Alzheimer's, different.
yeah.
So you can actually, with this technology, with this RNA sequencing, so to speak, you could alter, or maybe even like CRISPR, cut it out. So Alzheimer's becomes a thing of the past for human beings.
Well, I think this is. I think RNA therapy is actually a really great strategy uh, to help cure disease. Probably about 15, 20 years ago, it was all about stem cell therapy. And there's still really good work going on in that field. But the idea there was, we can sequence your tumor, or we can, we're probably not going to be able to take a brain sample from you because, you really can't do that to it. The brain makes up who you are. So you can't just go in and biopsy somebody's brain just for the heck of it. Okay. That also makes things a little tough, right? But, ideally, we get the information about what's wrong with you, right? Or what's wrong with the, causing the disease. Okay. So kind of like, what's really the next step that, we go there. And, idea is that you have to really fix the lesion in the DNA. And stem cell therapy or gene therapy originally came about by the understanding that there's only going to be a few cells in your body that you really need to target, right? So if you look at your hand or you look at the skin, right, all those cells on the top layer of your skin, they're all dead, right? So if you have a skin cancer and you try to target the cells on the top of your skin, I mean, it's a waste of time, right? Because they're just going to die in a couple of weeks. You have like a very few layer of basal cells that actually proliferate and repopulate your entire epidermis, okay, all across the skin, right? It's a different kind of organ, but the same thing happens in liver, same thing happens in your kidney, right? There are tissue-specific stem-like cells that repopulate the entire organ. So if you have cystic fibrosis, or you have Huntington's disease or something of this nature, we know the genetics, but you have to get a therapy in there to fix the mutation in the stem cell. So that's what kind of drove the field for the past 15, 20 years, right? Get a copy of a good gene in the stem cell and let that stem cell proliferate, grow, and repopulate the rest of the tissue with normal cells. Great. But the problem with that is, cell-based therapy is still just really kind of tough, I gotta say, to get that message into the right place at the right time. Kind of hard to do, and kind of what happened from that was it became less about targeting or sort of cell-based therapies necessarily, and the same challenge came about, which was delivering your entity, whatever it is, to the organ site. So it's still an issue of the therapeutic. Like in cancer, we just still treat people, a lot of patients with dumb chemotherapy, right? I mean, it's just what we do because it's metastatic cancer, and we've had these therapies around since the 70s, and they still work pretty effectively. I mean, if you look at somebody who has glioblastoma, they're still treated with TMZ basically, maybe some modifications, but for the most part, it's the same treatment. So we've made scientific advances, but we haven't really made good therapeutic advances, I think, in that field in the past 30 years. All right. So my point there is just that it's all about delivery, and it didn't matter if you started working in stem cells or you were interested in tissue engineering, or if you were somebody like myself interested in RNA as a therapeutic. It's still about trying to get your therapy to the right place and the right dose that's going to actually cure the disease.
I like been the actual issue with stem cells. They knew
Correct.
they work. They knew they proliferate, as you said. But they have a hard time delivering them at the right place at the right time.
It's a cell-based therapy, so it's very invasive also. I mean, you're not going to inject, you're not just going to take some stem cells and inject them into the blood intravenously and hope that they get to your skin. I mean, you have to inject the organ with the actual stem cell at some point. So if you have, trying to repair a liver, it's kind of an invasive procedure, I guess. And what happened because of that was we had iPS cells, and the idea there was we could take a skin cell and we could make it into a stem cell, and then we can then grow the stem cell and differentiate it back to a normal state. Like we can correct everything in a petri dish, basically, and then take those cells and put them into a patient. And that's still going on, and there's actually some really good work with that. I think that field lends itself to liquid tumors predominantly, like a lot of T-cell lymphomas and things. There's really some really great work by a few clinical investigators that really push that. I think it's tougher for the solid tumor field to take that strategy. And what we've kind of fallen back on, and what I just really preach more is, RNA as a therapeutic is great. I mean, it's injectable. We could just put some smart chemistries on the RNA molecule itself. It can be forced to go to certain tissue types. And then you can almost think about RNA delivery as a drug that given by pharmaceutical companies, right? Pharmaceutical companies are really good at making small molecules, right? Small drug compounds. They have libraries of ton of different analogs of all of the hormones that are out there, of all of the ATP blockers that are out there, all of the receptor tyrosine kinases. They have thousands and thousands and thousands and thousands of drug molecules. But we could do the same thing in the RNA field. And then a patient doesn't have to maybe undergo also a stem cell transplant, which is a little tough because if you have to get rid of the bad stem cell and then also replace it with a good stem cell. And some people who have certain leukemias, part of their treatment, they get a stem cell transplant after they get some chemo. But it's not really common in the solid tumor field to do that.
Well, Brian, let me interrupt because we're touching on something that's real common in the stroke world, and that's that
Okay.
they are really kind of making big the stem cell treatment may be a way to minimize the effect of stroke. They can't stop stroke, one understand, but they can minimize, lose like a therapy, like you're saying. But do you agree with that?
I mean, I don't know. So I'm just kind of delving into this field so much. So I'm not going to make any formal comments about which therapy may be the best strategy going forward. If they're going to, I would just say that if they're going to use a stem cell strategy, it could work. I don't know necessarily. They would still have to have that cell sort of be homed to wherever the lesion was. So if it's somewhere in the brain, then it has to go to the region of region of the brain that was damaged by the stroke. I mean, which is going to be the same challenge a little bit with RNA, but RNA is not a cell. It can get passively taken up into multiple different cell types. And who knows? We just published a paper about this, the role of microRNAs and other non-coding RNAs as regulators of inflammation in the brain, which is a lot of what I'm talking about also could be that RNA could help ameliorate the symptoms and the side effects of something like a stroke. Like Alzheimer's is thought as genetic, but also neuroinflammatory. Not the neurons themselves that are getting damaged, but the microglia, the astrocytes, all of the cell types that help support the neuronal cells, like live and function normally, they actually get damaged during those disorders. So the same thing could also be happening during stroke, right? You have leaky blood vessels. You then have immune cells that are not supposed to be there. It's causing kind of an inflammatory state, right? And so we could have some anti-inflammatory RNA-based therapies that could do the same thing. They wouldn't necessarily cure necessarily, but what we've learned in the stroke world, it sounds like it's sort of a repair system anyway. Like after you have the lesion happen, it's going to take some time for your body to heal from that, get rid of any leaky blood vessels, things of this nature, and try to restore some of the damaged tissue with functioning healthy neurons.
All right. Well, the reason I bring that up, I'm going to bring right here on my show, Sherry's last comment was she's looking into a clinic locally, and she's in New York and Niagara Falls. But she is looking in. She's thinking about getting some stem cell treatment therapy. What do you think of
that? She should try everything that would work. I mean, Cleveland Clinic is a reputable place, obviously, and there's some really great clinical scientists that are over there, and they probably have some really good strategies for using stem cell based therapies to heal stroke. I don't know if the therapy is directed toward the neuronal cell itself, or if it's more for blood vessel angiogenesis, something like this, or like I just said, something that helps it's a T-cell based therapy or something like this that essentially is mitigating the inflammation around there. There's a few different strategies for that. So if I would do some research and investigation, I mean, if you want, we can share contact information at the end of the show. I can follow up and see exactly what kind of therapy they're talking about over here, and kind of explain to her what's going on.
That would be really awesome.
From an expert view down to like a layperson's view about like what they're trying to do in that strategy. So like I said, it's not necessarily that stem cell therapy is a bad thing. Sometimes I advocate for the RNA therapy approach, because I think there's things we can do, and there's things we can treat for that make it a little bit superior in some cases to stem cell therapy.
Well, Brian, I think stem cells, I'm gonna say from a layperson, that you're rejecting it. It does solve the problem, but it really is a lot more complicated now. But I'm hearing you. I want to make sure I lead this conversation. The RNA inside of every human being is a messenger. It tells. What it's telling, it's a messenger gene, right?
Yeah. So the RNA that we're talking about actually regulates that message. So it's there's different ways in which a gene can be expressed, and that's the complicated thing. Like when we like how you're thinking about it and the way it's been explained to us, I guess, or to everyday non-academic individuals. It's essentially like, okay, DNA makes RNA makes protein, and that's the way the system works. And then the problem with that is just life is just complicated, and things regulate and bump into other things. Right. It's just how it is. So the messenger RNA is still the carrier of information that still exists. It's still, that's its job. The DNA doesn't leave the nucleus or anything like that. But the microRNA, the small RNA, is the thing that actually starts regulating that message. So that's where it's like an RNA language. So it's like a while back, some people thought it was just an RNA world, right? Like there was no DNA around. It was just RNA interacting with RNA, and maybe even RNA made protein. They didn't even need DNA. So I don't know if that's, it's kind of a cute little hypothesis, but it's really about that small microRNA cannot just regulate one messenger RNA. It could regulate the abundance of a number of messengers. And the cool thing about microRNAs is this stuff was found in from the developmental biologist side of things. So in C. elegans, you would have, it's a small nematode worm, and as it grows up, it goes through different larval stages. It goes through molting, basically. And what they found is that some small RNA, actually, that was the big thing. That was the surprising thing. That's actually what set kind of this field apart, was they found these worms that just were not going through the right larval stage. They just weren't developing right. And they did some reverse genetics, and they basically were trying to find the genetic region that was causing this problem. And they would do all these tests, and they would run things on, we call them blots. Basically, we can probe for the gene area, and then we can expose the thing on film and see how abundant some gene region is. Don't worry about that so much. And I'm pretty sure this is how, I wonder how this postdoc or grad student felt back in the late 90s. Was looking for this genetic region and figuring out that it's, it's like less than 100 nucleotides long. And if anybody knows anything about how long a protein is, you're thinking to yourself, okay, the codon is 3 nucleotides long. So every 3 nucleotides you have of DNA sequence, you make 1 amino acid, and that amino acid it's a building block for a protein. So I'm like, okay, so the person's probably thinking, now I'm down to like 60 or 70 nucleotides. So maybe a small peptide is being made that's causing this problem. But when they started doing northern blots, they found that it was an RNA sequence. And I think this is the lineage and the lethal genes that are in C. elegans that they found to do this. And they found that was eventually turned out to be a microRNA, a small little RNA, controlled or even prevented an entire organism from developing at the right time. So it's not like this is just random stuff happening, right? It's not like just one microRNA is just targeting a bunch of genetic information at will, right? It's sort of there are at some level that may be happening at very low level, but the real true microRNAs that are regulating a biological process probably have been evolutionarily conserved over billions of years. And so they are going to be regulating cell networks, essentially. So like we found one, like MIR34, basically regulates a whole bunch of pathways that the normal protein P53, a bona fide tumor suppressor, actually regulates. So it's a coordinated effort. It's not like these microRNAs know what they're really doing, right? It's just, they've been selected for over time because they sometimes have some survival. They provide some kind of survival advantage to the organism.
Right.
So these kind of genetic, things are just maintained over time. So you find these things, like, we won't get to know what happens until you study every possible connection. And there's just a bias. Some of these microRNAs are just biased to regulating, a lot of MAP kinase signaling pathway stuff. Sometimes some microRNAs just tend to regulate a lot of hormone pathways. It just happens to be what's this, the sequence of the microRNA and the sequence of the gene that is targeting. So those are the ones we're trying to find, right? Because those are the tried and true pleiotropic actors that are going to be helpful for us to restore normal cell function as a therapy. So we want to take advantage of that. We like the fact that those microRNAs target many different things.
I mean, I want to make sure I understand. We're out of time. We're running out of time. But when
Yeah,
you say, you're saying cure, you're saying if you can find this and taught to correctly using a microRNA, you could cure cancer, Alzheimer's, any disease like that.
If the biology is right and the therapy, the chemistry is right to get it to the right place, yeah. There's companies that have already done this because microRNA is similar to. I don't want to get too technical, but microRNA makes imperfect pairing to messenger RNA. So it's small sequence. It's only 22 nucleotides. It's a very short sequence, and not all of that sequence binds directly to another message. There's some bulging that happens. So people get worried about maybe off target, meaning that microRNA can maybe target some other genes. The stuff that I'm talking about when they bring it into the clinic is they try to make that more what they call siRNA, and that kind of means more an exact match. So all 22 nucleotides essentially are engaged in targeting the actual messenger RNA. But they can do funky chemistries now on the backbone of the RNA and do all this stuff that you talked about, which was to actually edit the genome. AstraZeneca, I think, or a few other companies. I could probably look it up and send it out to to your audience. They've already done this for some rare tumors, or some rare neurologic disorders. It's a therapy that's already out there. It's clinically validated, done. It's a done deal. It's been treating patients with it now for, I think, a year or so. And the effects look pretty promising.
Great.
So,
hey, Brent, are we going to wrap up to 10 minutes over time? But, I want to thank you so much for being here. And I want everybody out there that may be over this live or in the replay. Brian's contact information would be in the description of the video. And he got a really awesome website. And definitely, he's here to be. You heard him. He's here to even help. You get a question, ask him, email him. And Brian, I want to thank you so much for me and thank you for being my podcast. Appreciate it. Thank you, brother. Appreciate it.
Oh, thank you for the opportunity. I think we were supposed to talk a little bit more about the executive experience.
Yeah, that's all
but
right. We'll have you on again when we come back.
Yeah, you can invite me on again. We can talk about the fun thing.
Well, thank you very much, Brian. Have a great day out. Thank you very much.
All right. Thank you very much, sir. Thank you.